السلام عليكم ورحمة الله وبركاته. Dear students, today inshallah we will start the histology by a subject known as cytology. First of all, what the meaning of histology? Histology is the science that deals with microscopic study of tissue. Why we said microscopic? Because I use either light microscope or electron microscope. If I use naked eye, it's anatomy. There is another word, it should be written here, that you are going to see normal tissue, not abnormal. Normal tissue, the meaning of the word histology. While if it's abnormal tissue, it is another subject called pathology. What the meaning of tissue? There are many cells aggregated together to form a tissue, and the tissue form organs, and the organs form system of the body. What are the common microscopic types of microscopes used in studying the histology? We have light microscope and electron microscopy. The light microscope, here is the eyepiece, and here is a tube, and these are the objective lens, and this is the stage that I put the slide on it, and this is the source of illumination, and this is the stage, and this is the gross and adjustment and fine adjustment, and this is the electron microscopy. It works by beams of electrons. What is the difference between light and electron microscopy? The light microscopy, the initials are LM, while the electron microscope, the initials EM. The source of illumination in the light microscope is the day or the light, electric light. Here we use many stains so I can see different colors. The maximum magnification that I can reach is up to 1500 times. If I need more magnification, I'm going to shift to the electron microscope. The examined field, I can see many cells. On the other side, the electron microscope, the source of illumination is the beam of electrons. The only used stain is the osmic acid, so if something stained with the osmic acid appears black, if it does not stain, it appears white, and in between are grades of gray. I can magnify more than 100,000 times. The field of examination is very small. I can see only one cell or a part of a cell. Let's ask ourselves what type of microscope that I examine such slide. This is light microscopy. Why? I can see many cells. I can see the different colors, the blue and the pink. And I cannot see details of the cell, so it is light microscopic photo. What about this slide? It is an electron microscopic slide. Why? I can see the field contain only one cell, and the stained either black, gray, dark gray, or light gray. And I can see details of the cell. I can see details of the cell. What are the common stains used in the histology? The common stains known as hematoxylin and eosin, and the initials are H and E. Here I'm going to tell you the basic of the stain. Hematoxylin is the basic stain. Its color is blue. It stains any acidic components, which as the DNA, the nucleus, and the RNA. This acidic component will be called the acid like something basic, so it is called basophilic. Philic means love or like, and appears blue in color. Again, if the something is acidic in nature as the nucleus or the RNA present in the ribosome, it will be stained with something basic, and this will be called basophilic and appears blue in color. While if something is basic in nature, like to be stained with the acidic stain, and this will be called acidophilic and appears red color. Again, eosin The eosine stain any acidic component. Eosine stain any basic component, and it is called acidophilic. Any basic component like to be stained with acidic stain known as acidophilic. Dear student, this is light microscopic picture of a section stained with hematoxylin and eosine. Why light microscope? I can see many cells. Why hematoxylin eosine? Because I can see all the nucleus stained with blue, either light or dark, and the cytoplasm either acidophilic or basophilic. The first subject we are going to study is known as cytology. I'm going to study the cell. Why I'm studying the cell? Because the cell is the structural and the functional unit of the living body. The cells have different shape and size. The cell is formed of nucleus and cytoplasm. I can never say it is a cell unless it contains nucleus. The cytoplasm contains three important components, which are the matrix, inclusions, and organelles. The organelles are permanent, usually present, always present, essential for a vital function, and they are specialized, do special work. So I can remember three words: permanent, essential, and specialized. The inclusions are temporary, non-essential, except some of them, which is myoglobin and hemoglobin. They are important inclusions. Most of the other inclusions are stored food as lipid and glycogen. While the matrix contains a solution of protein, carbohydrates, lipids, and enzymes. The organelles, either membranous organelles or non-membranous organelles. Membranous organelles as cell membrane, mitochondria, endoplasmic reticulum as rough and smooth endoplasmic reticulum, Golgi apparatus, and lysosome. The non-membranous either ribosomes, cytoskeleton. Microtubules as centriole, cilia, flagella, filaments as thin filament, which is actin, and other intermediate filament. First, we're going to talk about the cell membrane or the plasma-lemma, which is important for transport. By light microscope, it is difficult to be seen because it is very thin. If you want to see it by the light microscope, you should stain it with silver or pass. By electron microscope, the cell membrane appear as three lines, two dark lines, in between them one white line. This is called trilamellar membrane. Two dark and in between them one light. In order to know why the EM appears like this, you should study the molecular biology of the cell membrane. The cell membrane consists of lipid, which is known as phospholipid molecule, arranged in two layers, head directed outward and tail directed inwards. There is cholesterol in between the hydrophobic tails. This is a diagrammatic picture showing the molecular structure of the cell membrane. It is formed of lipid bilayer, the head directed outwards towards the outside of the cell and towards the inside of the cell, and the tail is directed inwards and cholesterol embedded in between in order to stabilize the phospholipid bilayer. The protein component, either we have intrinsic, which is either small or large. The large is known as transmembrane protein that transverse the length of the lipid bilayer, the peripheral loosely attached. The carbohydrate either attached to the protein, so-called glycoprotein, or the lipid called glycolipid, forming what so-called cell coat or glycocalyx on the external surface, which carries the receptor. Again, dear student, this is the phospholipid bilayer. Okay, and these the heads, and these are the tails. Heads directed outwards, and this is a tail directed inward. Here is the proteins, either transmembrane protein or peripheral protein. and the carbohydrate either attached to the protein called glycoprotein or attached to the lipid called glycolipid. What is the function of the cell membrane? The function is transport. We have many types of transport. First, we have the passive transport that passes without the need of energy, moves with concentration gradient, and it is subdivided into two groups: simple diffusion as water, ion, and gas, and facilitated diffusion moves with concentration gradient but through the transmembrane proteins as amino acids and glucose. Needs carrier. Active transport, this moves against concentration gradient as sodium potassium pump. All the time I say the sodium should go outside, so the concentration in the sodium in the outside is more than the inside. It enters passively and it got outside the cell actively. Bulk transport, active transport through which the large particle passes through the cell membrane. Here, dear students, this is the cell membrane. Okay, these are the transmembrane proteins. Here's something outside want to enter. If the moves with concentration gradient, we have either two types, simple and facilitated. Both of them passive, does not need energy. If I'm moving against concentration gradient, I need energy, and this is called active transport. Active transport either endocytosis. The endocytosis, something enter from outside to inside. We have either phagocytosis. I let something solid to enter inside the cell. The cell sent pseudopodia that fuse together, entering the solid particles. Part of the membrane will become inwards with it known as phagosome. And I'm going to show you a picture right now. Pinocytosis, fluid enter by pinocytic vesicle. Here, dear student, this is the cell membrane, and this is the solid structure that want to enter. The cell membrane sends pseudopodia, and the particle enter together with a part of a cell membrane, and this is called the phagosome. This is called phagocytosis. While pinocytosis, fluid enter inside the cell membrane will make an invagination, and the cell membrane together with the fluid known as pinocytic vesicle. What the meaning of exocytosis? A process by which the cell extrudes residual products. The secretory vesicle fused with the membrane and its content are discharged. This is a diagrammatic picture, dear students. This is the endocytosis and here is the exocytosis. Endocytosis, part of the cell membrane will enter together with the content, while in exocytosis the vesicle contain the undigested material and want to get outside the cell. The membrane will fuse again and the content that I do not want will go outside. As you can see here, the part of the membrane that enter is the same that is here. I didn't lose any membrane. What is the function of the cell coat? What is the function of the cell coat? As we said, the cell coat is the glycolipid or the glycoprotein that present on the surface of the cell, and they contain receptors. So the first function is selective permeability. The cell coat is important for protection, adhesion and recognition. Adhesion, cell junction occurs at this part of a cell. Recognition, each cell send part of its antigen to the surface in order that the immune system know that this cell is related to itself. Avoid doing any autoimmune response against it. We have cell membrane modification, either microvilli to increase surface area, cilia to move particles in one direction. Stereocilia, it is long microvilli as present in the epididymis to increase the surface area for absorption. The last function is conduction of excitation wave for nerve impulse to pass to and for the muscle to contract. The organelle that is responsible for energy production is called the mitochondria. As you see here, the mitochondria is formed of two membranes. The outer membrane is smooth and the inner forms what's so-called cristae to increase the surface area. Here, the enzyme needed for energy production are present on this cristae. And this is the matrix, and the membrane, the space between the two membranes is called intermembranous space. It cannot be seen by the hematoxylin and eosine. It needs special stain known as iron hematoxylin and Janus green. Appears as granules, filaments, or rods. By EM, the outer membrane is smooth while the inner membrane form cristae. Cristae that increase the surface area. Shelf-like folds. To increase the surface area to respiratory enzymes. The matrix fills the cavity, contains lipid proteins, RNA, DNA, and respiratory enzymes, catalysts as calcium and magnesium. Because the mitochondria contains DNA, it can self-replicate and increase its number. The size and the number of the mitochondria variable differ from one cell to another. Active cell contain many mitochondria up to 1000 or 2000 as in the liver cell. Inactive cell as lymphocyte contains small amount of mitochondria. The size is variable as, for example, when a cell has a cilia, the mitochondria is present at the apex of the cell. Function is energy production, and it can self-replicate as it contains its own DNA. Endoplasmic reticulum, we have two types. Endoplasmic reticulum are system communicating, membrane-bound communicating together, either rough or smooth. Rough due to presence of the ribosome, smooth due to absence of the ribosome. The rough called granular and the smooth called non-granular. As you can see here, it is a membrane-bound intercommunicating channels. About the rough endoplasmic reticulum present in protein-producing cell, as the ribosome is the factory that produces the protein. And this protein will be accumulated in the rough endoplasmic reticulum, as we're going to take right now the function of the RER. By the light microscope, it appears basophilic. Why? As the ribosome is acid-like to be stained with an basic stain, so it is basophilic and appears blue in color. By EM, it appears as membrane-bound sheets or cisternae intercommunicating together, ribosomes present on the surface attached to the rough endoplasmic reticulum by the large subunit. Here is a cisternae. This is a diagrammatic picture, and this is a cisternae for the rough endoplasmic reticulum. Here are the ribosomes attached to the rough endoplasmic reticulum and form the protein. What is the function of the rough endoplasmic reticulum? Storage of the protein synthesized by the ribosome, segregation of this protein, cutting it, segregation, and initial glycosylation. This is an EM picture of rough endoplasmic reticulum. Here are the cisternae, regular, on the outer surface, electron-dense granules, which are the ribosomes. Again, this is an EM picture of rough endoplasmic reticulum, regular cisternae, membrane-bound on the outer surface. There are ribosomes. There is an EM picture of a protein-producing cell. This is the nucleus, and this is the rough endoplasmic reticulum extending from the nuclear membrane to the cell membrane, and these are the mitochondria. Functions of the rough endoplasmic reticulum: accumulation of the protein produced by the ribosome, segregation of the protein, initial glycosylation by adding glucose, packing and releasing by budding from the rough endoplasmic reticulum in a form of transfer vesicle. Some of the enzymes produced inside the rough endoplasmic reticulum are hydrolytic enzymes, so their presence inside the tubules of the rough endoplasmic reticulum protect the cytoplasm from these hydrolytic enzymes, and as well as important for the intracellular transport. Important for intracellular transport. Here again, this is a diagrammatic picture show the relation between the rough endoplasmic reticulum Golgi apparatus. This is the rough endoplasmic reticulum protein produced here the transfer vesicle budding from the rough endoplasmic reticulum and go to the Golgi apparatus. Smooth endoplasmic reticulum, here intercommunicating membrane-bound channels connected together, branch and anastomose, and does not contain ribosomes on the surface. Okay. It's present in steroid-secreting cells or fat-secreting cells in the liver and endocrine glands. YLM is not stained by the hematoxylin and eosin, but when abundant appear acidophilic. Why? Because it is a membranous organelle that does not contain ribosome on the surface. These membranes contains amine group in the proteins, in the protein parts of the molecular structure of any membranous organelle. We have protein. The protein contains amine group. The amine is positive charge, like to be stained with a negative charge stain, which is the eosin, so it appears acidophilic. By EM, a membrane-bound tubules or vesicles with no ribosomes on the surface, and they branch in anastomose. This is EM picture. As you can see here, they are irregular because they are branching and anastomosing, and the outer surface does not contain ribosomes. It is well developed in fat and steroid secreting cells. The function is synthesis of lipid and steroid hormone, hydrochloric acid formation in the stomach, which is important for digestion, muscle contraction by release of calcium pump. It stores calcium pump during muscle contraction. The calcium is needed to bind to certain protein to uncover a part in the actin filament in order to react with the myosin and lead to contraction. Detoxification of drugs and hormones in the liver, storage and lysis of glycogen in the liver, and intracellular pathway again from the RER to the Golgi apparatus. The Golgi apparatus is the secretory apparatus of the cell. By hematoxyl lysine does not appear. This is diagrammatic picture. This is a protein producing cell, appears basophilic. Here an area that appears pale. This is the Golgi apparatus that not stained by hematoxylin and eosin, so it will be called negative Golgi image. If you want to stain the Golgi apparatus, stain it with silver stain, it appears brown, granules or fibrils. In the nerve cell, the Golgi apparatus present around the nucleus, so-called perinuclear. In the secretory cells, appears in the apical part of the cell. Here is the Golgi apparatus. Appears as 3 to 10 flat parallel intercommunicating circuits forming what's called stack. They have two surfaces, convex and concave. The convex or the immature, the transfer vesicle arises from the rough endoplasmic reticulum fuse on this side, receive the transfer vesicle, and the concave or the mature release the secretory vesicle and lysosomes. What is the function of the Golgi apparatus? Concentration of the secretory product arises from the RER through the transfer vesicle. Modification of the protein on this transfer vesicle by adding to it carbohydrates and sulfates. Discharge the secretory products, either enzymes or hormones. Formation of lysosome and maintenance of the cell membrane. Here is their diagram show the relation between the rough endoplasmic reticulum and the Golgi apparatus. From the rough endoplasmic reticulum, transfer vesicle arise, and here the convex face of the Golgi apparatus receive this transfer vesicle. From the mature face, secretory vesicle or lysosome is produced. secretory vesicle fuse with the membrane of the cell membrane, and the content moves out by exocytosis. Here is the function of maintenance of the cell membrane as we add part of the membrane to the actual cell membrane, so it's the function maintenance of the cell membrane. This organelle is the lysosome, which is the digestive apparatus of the cell. It arises from the function of both rough endoplasmic reticulum and Golgi apparatus, as the hydrolytic enzyme is formed inside the rough endoplasmic reticulum and move in the form of transfer vesicle and go to the Golgi apparatus, in which the concentrated beta modified and released in the form of a lysosome. The number of lysosomes increased in phagocytic cell. It is not seen by the hematoxylin and eosin, seen by histochemical test. The lysosome has enzyme known as acid phosphatase. If I can detect the acid phosphatase, so this cell contains lysosomes. By EM, we have two types of lysosome. Primary lysosome, which is newly formed, arises from the Golgi apparatus. It appears homogeneous, surrounded with single membrane, does not yet enter any digestion. While the secondary lysosome contains digested material and undigested material, it appears as heterogeneous, formed of fusion of the primary lysosome with any phagocytosed particles. Here is a diagram showing the relation between the rough endoplasmic reticulum, Golgi apparatus, lysosome, and cell membrane. The rough endoplasmic reticulum produces transfer vesicle that fuse with the Golgi apparatus. The rough endoplasmic reticulum forms the digestive enzymes, and these digestive enzymes will be modified, concentrated, and released in the form of primary lysosome. Fusion of the primary lysosome with solid particles, this is known as heterolysosome. With pinocytic vesicle, this will be multivesicular body. Fusion of the primary lysosome with old substrate as old mitochondria, known as autolysosome. Again, types of secondary lysosome: heterolysosome, which is fusion of the primary lysosome with phagosome. Multivesicular body, fusion of the primary lysosome with pinocytic vesicle. Autolysosome, fusion of the primary lysosome with destroyed organelle as the mitochondria. After digestion, there will be residual body. This is a membrane containing undigested material that opened by exocytosis. Again, diagrammatic pictures show the relation between the rough endoplasmic reticulum that release transfer vesicle contain the hydrolytic enzyme, enter the Golgi apparatus through the convex phase. In the Golgi apparatus, modification occurs in this enzyme, released from the mature phase in the form of primary lysosome. Primary lysosome fuse with either old mitochondria, forming autolysosome, multivesicular pinocytic vesicle for multivesicular body, phagosome, heterolysosome. After digestion, residual body, it's a membrane containing undigested material opened by exocytosis. Fate of the residual body: discharge of the material outside the cell by exocytosis, or it might accumulate in over years in aged cardiac and nerve cell, as the nerve cell and the heart cell are not dividing cells, they are mature end cells. As the cell increase in age, this residual body increase their number in such cells. What is the function of lysosome? Digestion of solid fluid or old organelle, digestion of the whole cell after death, help in fertilization as the ova is surrounded with a thick shell or thick wall. Millions of sperms contains in their head hydrolytic enzymes try to help one sperm in order to fertilize the ova by hydrolyzing this thick shell. Release of the thyroid hormone. The thyroid hormone produced in the thyroid gland produced in a large amount, and it's stored in the form of inactive form. When the thyroid hormone is needed, lysosome break the inactive form and change it into active one. The nucleus. Any nucleated cell, any cell contain nucleus. The nucleus is formed of nuclear membrane. It's formed of two layers, outer and inner nuclear membrane that fuse together in areas known as the nuclear pore. There is the chromatin, which either hetero or euchromatin, nucleolus, and the nuclear sap. The nucleus present in all cells except the RBCs and the platelets. The numbers of the nucleus usually 1. In the liver we might have 2, and in the skeletal muscle multiple nucleus. Position may be central, basal, or at one side eccentric, may be rounded, oval, or segmented. Stained basic stain appears basic. Why? Because it's acidic in nature, contain the DNA. The DNA acid like to be stained with a basic stain known as basophilic and appears blue. The nuclear membrane or the nuclear envelope formed of two layers, outer and inner. The outer layer contains ribosomes or rough endoplasmic reticulum continuous with it, and the inner nuclear contains the peripheral chromatin on the inner aspect. It appears basophilic line. By EM it is double walled, connected together at the nuclear pore. The outer membrane covered with ribosomes or rough endoplasmic reticulum, and the inner lined with peripheral chromatin. Nuclear pore at which the outer and the inner membrane fuse together. Chromatin, it is formed of DNA and histone protein. Appears basophilic particles or threads. The chromatin either euchromatin or heterochromatin. The chromatin carries genetic information, tell you how certain protein is formed. If I want to synthesize this protein, I'm going to copy an message from the DNA on the messenger RNA. So I should extend this chromosome so it will appear pale, and this is called euchromatin, as it contains active genes for having a copy on the messenger RNA to start the protein synthesis. Heterochromatin, this chromatin is condensed and contained in active gene, and by EM appears dark. The heterochromatin present arranged either on the inner nuclear membrane called peripheral chromatin, groups forming chromatin islands, or associated with a nucleolus known as nucleolus associated chromatin. The nucleolus is rounded, basophilic structure. This is the EM picture. Okay, rich in RNA. It is non-membranous. It's formed of dark areas containing the DNA and the RNA, light areas containing the nuclear sap. The nuclear sap is a colloidal substance between the chromatin and the nucleolus. It's important for transport of the RNA through the nuclear pores to the cytoplasm, as this RNA is needed for the formation of ribosomes. What is the function of the nucleus? Carries genetic information, formation of all types of RNA, rRNA, messenger RNA, and transfer RNA, and it's important in protein synthesis. In a protein-forming cell, the cell appears basophilic due to presence of rough endoplasmic reticulum. By EM, I'm having three important organelles, RER, Golgi apparatus, and mitochondria, and the nucleus appears pale due to the presence of euchromatin. By light microscope appears dark basophilic and the nucleus pale with visible nucleolus. We finished the membranous organelles and the nucleus. We will have a hint on the non-membranous organelles. These are organelles that's not surrounded with membrane and includes the ribosomes, which is important for the formation of the protein. The rRNA is formed inside the nucleus. And the protein parts formed in the rough endoplasmic reticulum that pass to the nucleus and fuse to form the small and the large subunit, then go out to the cytoplasm in order to produce the protein. They are RNA, so stained with hematoxin and eosin. They are acidic in nature, so they are stained basically with and it's called basophilic and basic stain, and appears basophilic and appears blue in color. The ribosomes either present free or attached on the outer aspect of the rough endoplasmic reticulum. The function protein formation. If it is free, it's used to form the protein needed by the cell as glycolytic enzymes. So this is the free ribosomes, small and large subunit attached by the messenger RNA. They form protein that's used by the cell. Attached ribosomes present attached on by the large subunit on the rough endoplasmic reticulum and produce protein. This protein will be released outside of the cell as hormones and enzymes. The cytoskeleton is a network of protein molecules needed for keeping the shape of the cell and help the cell to move and to make process as pseudopodia. It includes microtubules, filaments, and intermediate filaments. This is an EM picture of the cytoskeleton. Data show slides. This is an EM picture of two adjacent cell membrane. Here you can see the trilamellar appearance. Two dark lines separated by one light line. Here is the molecular structure of the cell membrane. This is the phospholipid bilayer, and this is the transmembrane protein. And this is the carbohydrate attached to the protein, glycoprotein attached to the lipid, glycolipid. And the protein, either the extrinsic protein or the integral protein. This is an EM picture of the mitochondria formed of outer membrane, which is smooth, and the inner membrane forming cristae. This is an EM picture of mitochondria and rough endoplasmic reticulum. You can see the flattened cisternae here, which are parallel and covered with ribosomes. Here is an EM picture of the Golgi apparatus, which is formed of stack of 3 to 10 sacules, two surfaces, convex and concave. The convex transfer vesicle goes to it, while the concave arises secretory vesicle from it. Here is EM picture of rough endoplasmic reticulum in which it is regular cisternae, and you can see the ribosomes present on the outer aspect. Here is smooth endoplasmic reticulum, irregular tubules. I can see cuts longitudinal NTS due to branching and anastomosing tubules and no ribosomes present on the surface. Here, dear student, this is an EM picture of secondary lysosome, different types of secondary lysosome. As you can see here, this is heterolysosome, appears heterogeneous. Primary lysosome fused with phagosome. This is multivesicular body, as well appears heterogeneous. It's formed of primary lysosome fused with pinocytic vesicles. Again, this is heterolysosome. You just say it is an EM picture of secondary lysosome. This is an eochromatic nucleus. I can see most of the nucleus eochromatin, pale, and this is prominent nucleoli. While this is heterochromatin, in which most of the chromatin is heterochromatin. Here is the peripheral chromatin and islands. This is heterochromatic nucleus.